Welcome back to the Merxin Ltd podcast. Today I have the pleasure of having a conversation with Gary Pitcairn. Hello, Gary.
Hello, it's nice to be here.
Thank you for coming to Central London for a conversation about the inhalation drug delivery. So it's fairly relaxed. What I would like to know is who you are, what is your career, what is your view of inhalation drug delivery, what is the future? So let's start with the first question. Can you introduce yourself to the people who don't know about you, and or who don't know you, I should say, and will be watching this podcast? Who are you, Gary?
Okay, thank you. So, Gary Pitcairn, I am currently a consultant helping companies develop new respiratory products. I have approximately 30 years of experience in working with different companies to develop products. So back in the very early 90s, I started at a CRO and did lots of in vitro and in clinical studies there. Then after a number of years, I joined Big Pharma, and I've been at Pfizer, Mylan and AstraZeneca before deciding to dip my toe into the waters of consultancy. So I very much enjoy what I do. I think of myself as a problem solver. I describe myself as an interface worker between the pharmaceutical development, the device development, and the clinical application. So a lot of what I do is help people take molecules through to the clinic and then beyond thereafter. And I'm happy to say I really quite enjoy it.
And of course, you're very famous in the inhalation drug delivery space because you're the chairman of DDL. How long have you held that position?
It's 20 years now this year. So 20 years have been on the committee. Probably about 13, 14 of actually being the chair. Yeah. So
quite a piece of work. Yes, quite a lot of experience there. We will talk about that in a minute. There was some interesting movement, as noticed in my email in the last few weeks, for young researchers at DDL and so on, seminars over the summer. So I want to quiz you about this. What was the name of the CRO you started at?
it was a company called Pharmaceutical Profiles.
Oh, yes, Nottingham.
Working with Steve Newman, who, of course, is, I think, properly famous.
Yes.
I kind of, he took me under his wing. I did a part-time PhD there over a number of years.
So was that at Nottingham University as well?
Yes. I was at Nottingham, and then I joined Pharmaceutical Profiles as I thought as a stopgap job to earn some money before doing a PhD in chemistry. But then I got into the world of inhaled products. I found it fascinating, and here I am 30 plus years later.
This is amazing. We all probably started at the same time, sort of doing our PhDs in the 90s or early 90s. So you've been a long time in inhalation drug delivery. You mentioned Pfizer, you mentioned Viatris, I think. Were you at Viatris?
Yeah.
And then AstraZeneca. In that long career, what has struck you? What are the couple of things you will remember of your career? Defining moments either in the industry or in the companies you're working with, or some of the products you might have developed. Couple of milestones you think were really defining.
So going back to when I was at Pharmaceutical Profiles, I did a lot of work with a company called Boehringer Ingelheim, working through different prototypes of soft mist inhalers.
Okay.
And the initial ones were quite big and clunky, doing lots of in vitro work, but then taking them into clinical studies, different drugs to show that they do indeed deliver. That was really interesting because that was something quite different from the sort of the turbohaler and the standard meter dose inhalers with or without a spacer that I'd been doing. So that was one moment that it felt like that was something properly new. The other one was working still in the same company with Inhale at the time. They went on to become Nectar on developing inhaled powders for systemic delivery. So I worked on insulin and a number of other different molecules. And it was interesting then because that was, of course, an ongoing project when I joined Pfizer. And so that was perhaps the next defining moment when that decision to stop for various reasons was taken. It felt up to then the world of possibility was opening, new devices, opening up different opportunities, sort of a move to systemic therapy. And then it felt a little bit like the rug being pulled from underneath your feet at that time. So those are the ones that ping to mind.
So Boehringer Ingelheim SMI and Pfizer Exubera,
Yeah,
essentially,
yeah,
inhaled insulin. Of course, it wasn't the death of inhaled insulin because
no,
mankind eventually developed and launched its own product, and it opened certainly the eyes of everybody of what could be done with formulations and biomolecules. Um, I mentioned something earlier about DDL and young people. So, looking back at your career, if you were to give an advice to a young person starting now, like young Gary 30 years ago doing his PhD, what would you tell him and the other students today?
that is a good question. I think the world has changed because going back when we started, there were just a small number of large pharma companies, and you were probably fairly comfortable once you were within that company of having a long and industrious career. That world has changed, of course, and now I think there are much smaller companies. There's a lot more change. I would advocate, and I've had this conversation with people that I've mentored, to build in a breadth of experience, not necessarily just try to focus in an area of expertise in one particular, but to build that breadth to make yourself more useful across how a company performs, to be more aware of how organizations function. So to be a bit more robust to change, because the change comes whether you want it or not. And it has surprised me in the past. I think I'm a lot more resilient to that now after I think I've been made redundant 3 times now in total. Um, but I think it helps if you have some breadth to fall back on. You are more appealing because you're not a one specific thing. You can clearly, you've got a track record of applying yourself to different problems in different situations. I think that would be helpful.
I think this is very good that you put your finger on redundancy because the industry has changed a lot and keeps on changing. And there's a lot of movements. And at some point, we've all been at sites that have closed down. AstraZeneca closed down Charnwood, Novartis closed down Horsham, where I was associated with. You had your own experiences. And of course, we led to be afraid of those events. And instead of embracing them and said, well, it is part of the change, do not be afraid. And of course, we're all afraid. And I'm glad you mentioned it because we need to tell people do not be afraid. There will be wonderful opportunities. You manage to bounce back every time. I managed to bounce back. Others have managed to bounce back because we are resourceful, we have talent, and there are opportunities out there. And there are exciting product and industrial opportunities. And for those who are in academia, there are also opportunities out there throughout the world. And I think it's a very hopeful message. I'm glad you mentioned it because actually there are so many ways to be having fun, but not being stuck in the same company forever. So
Exactly. It's difficult though. I mean, one has to embrace the uncertainty, but you know there is something coming and you can apply yourself to that. And I still speak to a number of people who are going through it because, of course, it is now a constant. Lots of the companies are only funded for a short period of time up to a certain milestone, and then it may go ahead, it may not. So I think it more and more it's common. So I think becoming okay as you can with that sense of uncertainty because you know something, it will open another door. It's the way to proceed.
So we're talking about change here and embracing it. What do you think is changing in our industry? Either therapies, molecules, the way companies are organized, maybe the way clinical trials are changing. We're discovering that a lot of clinical trials happening in China, which go much, much faster than anywhere else in the world, except maybe in India, where it might go very quickly too. So what are the changes you're noticing that are important to you?
Well, I think there's been a noticeable moving away from smaller molecules to larger molecules.
Mhm.
I think really what we're seeing now is the sort of the need and the development of better dry powder inhalers, better liquid inhalers, nebulizers, soft mist inhalers, to really provide for the molecules that are coming through, the molecules that are in the portfolios now. And these are principally biologics. And so it is very much focused on that area of these larger molecules for sort of treating a whole range of perhaps non-traditional indications. We talk about cancer, we talk about
Mhm.
infection, and other things, and I think that's the direction of travel. And I think that does it does cause changes, of course, as the industry has rebalancing what the need is. We've had I guess recently, and there's still some ongoing work about the replacement of propellants for the metered-dose inhalers. But then that therapy is sorted in the sense of there is good and very efficient treatment, if you like, for the topical treatment of asthma and COPD. Now the moves seem to be going to other modes of treatment to perhaps more efficiently treat things. And so I think it's interesting to watch. That tends to be, I think, driven a lot by smaller companies, more biotech, um, with sort of reaching out and getting funding, and then linking up with people who are developing a molecule concept with, well, how do we progress this? What device do we put it in? So I think it's smaller groups forming for a period of time and perhaps working together rather than the bigger pharma, the world that we knew of years gone by.
Yes. So so that two questions from me. First of all, you mentioned smaller companies. Any names you would like to mention of companies you're following by any chance? You think, oh, that's interesting therapy. That's a bit new. Or
Yes, I mean there's a number of them. There's keep an eye on one of the I mentioned the biologics, but of course the delivery of RNA, for example. There's different people developing different strategies for how is that to go ahead. And of course, it's by no means that we've done this. It's not done and dusted. This is in development. It's still, I think, really needs to be proven. But a company like Ethris, who've sort of developing mRNA, I was reading a fascinating story the other day of delivery of via inhalation to treat cardiac events by a
what was the
company,
name of the company?
Nanophoria
Oh,
in
okay.
Italy, and they've recently secured lots of funding. And it's based on, I think, some really, really good sound science that's been done in academia and then has developed. And the concept is beautiful because they have a peptide which they put in these inorganic nanoparticles, calcium phosphate nanoparticles, which you can't easily deliver, of course. And they went, well, that's fine. We'll embed it in a microparticle. We'll deliver that to the lung, rapid dissolution, and delivery that way. I mean, that's clever, and I think that's also a potential way of delivering RNA as well. So those developments, in a way which you think, okay, this can be. It's not just a very small batch done in an academic unit. It's something which can be done. It's using technologies which are known to be scalable. You start to think, okay, well, this has, I think, has potential. Things like that which capture my interest.
Offline, you mentioned that one of the areas you're following is lung cancer and inhaled
Yeah.
therapies. Can you tell us more why and what's the attraction?
Well, it is. I mean, we all know, I imagine, people who have been touched by lung cancer, and so it's quite horrible. The idea of being able to sort of use our expertise or knowledge in inhaled therapy to potentially tackle that. Because the thing that caught my attention was we have drugs which work very well, but ultimately they're limited in how often you can give them and the doses you can give them for the dose limiting toxicity in the organ. So the idea is to, and then it was, well, but these tumors aren't just sitting as a big lump in the airway. You've perhaps only got a small amount of the tumor accessible. But nevertheless, there's been some really nice work done showing that you can actually get inhaled particles into these tumors, and it is effective. A company called Inhale Target, working with cisplatin, for example, inhaled, and they've done some really nice work showing that this is actually not only safer, but it's also efficacious. And that work is developing. It's now in clinical trials, and I love the fact that there is a potential to not replace conventional treatments, but as an add-on, as a supportive way to be able to perhaps give the drugs for longer within a safety profile to really maximize on their known efficacy. That seems to me a very nice application of, if you like, our technology to absolutely deliver upon a clinical need. So I'm, I read those papers with interest because you want that breakthrough moment where something is, past phase 3 and you think, aha, good, because this can only be to everyone's benefit.
So you're focusing on certain areas, and you're particularly following lung cancer. And we talked about the changes that have happened. What are you looking forward to in the next 5 years in the inhalation drug delivery space? What will, from what you know today, what is on your radar and you look forward to?
It, it is a good question. I would like to see some of these opportunities move successfully past into phase 2B, into phase 3. So really the safety and the dose has been properly established. These now look as though they're likely to become um, medicines which will be available to patients. That 5-year window may be a little bit challenging, but that's the direction of travel. I think there's other things. I get working with a number of different clients very early on, working through some of the preclinical, how do we demonstrate proof of principle, and of course the necessary safety studies. You become acutely aware of the number of animals you get through, and then some of the biologics you are required to do non-human primate studies. And so I think that the development of better in vitro models, better cell-based models, where you can get decision-making data, which perhaps reduces the number of subjects you have to use preclinically before going ahead. I think that would be very, very satisfying because and these are difficult questions and you have to do some of this work. But I think we've seen at conferences the development of better cell-based models which better mimic proper breathing to give you an indication of, okay, have you got target engagement? Yes, no. What are your indicators, the markers of safety, and actually coming up with meaningful data. That's a direction which I hope that these models can improve upon to minimize what we need to do preclinically. And as well as being, obviously that's a good thing in itself, but also for our industry, that reduces the cost and will reduce the time. And a number of companies I work with, they struggle to find the funding, for example, for the GLP studies, because we know it takes a large amount of material, it takes a lot of pre-planning, and it's very, very expensive to do those GLP tox studies. If you can reduce the scope of those studies, if you can reduce the time frame in which it takes to sort of get through that work, I think that would perhaps help us move more quickly to getting into the clinic. Whereupon a lot of what we do is based on predictions and guesswork and other aspects. We need to have, of course, the clinical data to be sure. If we could get there faster, I think that would be to the industry's benefit.
It's interesting you mentioned that because last week I was reading an article. I can't remember if it was The Economist or one of the industry publications devoted to clinical trials or general analysis of the pharmaceutical industry. And they were commenting on the fact that so many clinical trials fail and saying, why do they fail so often? And their conclusion was, it's probably the way the study is carried out. Population choices, protocol choices, spreading out the research centers, endpoints which are not flexible enough or are not reactive enough. The fact that the study is very fixed rather than adaptive. And so there's so many more molecules might be saved. And you see that all molecules that have failed certain clinical trials are then picked up by companies like Roivant and repurposed and rediscovered and reworked on, discovered. They have value, but clearly the clinical trial that was planned was not adequate. What's your view on that? Because do you think we could redesign clinical trials to improve the success rates by allowing more thinking and more flexibility and understanding of science?
I think so. I think there's an easy answer to this question, which I think is something that you've done as well, which is for inhaled products, is to have better control of how they're actually used in the clinic. I've been involved in many, many clinical trials where it's almost like handing over a fence. Here's the product, you have a protocol, go ahead and implement that. But the staff at the clinic aren't trained. They don't necessarily understand how to use the devices. Clearly, the volunteers may not know how to do it. Perhaps many of the patients don't. And I think building that training into how the conduct of the study with the devices is very, very important. So I've done that lots, and I think that greatly reduces the risk of a bust study by improving the confidence you actually manage to deliver your medicine. But that's perhaps an easy fix up in the front end. I think you mentioned about flexibility to adapt to what you see. I think too many protocols are written in a very fixed, we will do this in this order, not enough interim analysis, What do we do
That's what I was
next?
saying,
Okay.
interim, correct.
And being afraid, embracing some of that risk, but having a plan which allows you to flex around that. This is what we could do. This is how we could respond to try and maximize the benefit of the data that you're obtaining. I don't think it should just be a tick box exercise, a clinical protocol written in, if you like, carved in stone, which you will follow through to the end. I think that's a very that just doesn't work, does it?
Now I remember the source. It was a conversation on LinkedIn in one of the groups I moderate or I
Okay.
created. It's exactly the point that we're making, that you need to analyze more and don't simply go blindly to the clinical trial. You start and you wait for the end, because so many things go wrong, and you need to understand, to be reactive if you want to save your molecule. Because often the clinical trials have failed, not because of there are obvious lack of efficacy in certain molecules or a lack of benefits or toxicology, but there is also a problem with the protocol maybe itself and the population and the studies and the analysis. Related to that question about improving the outcome of clinical trials to have successful programs, what do you think is the role of in silico modeling or studies?
I think it can play a huge role in increasing confidence about outcome. We know that when we go into these trials, we make a number of assumptions about how the product is likely to perform. I think by having more data available, modeling different outcomes, different scenarios, different parameters to get a sense of where's the robustness, where does the risk sit. I think there's been some really nice work in this area. We're aware, of course, like FRI, the sort of functional imaging methods where you can start to build up a better understanding of how your product is likely to perform in the lungs of people with different levels of disease, different disease severities. I think that helps. But all the overall direction of being able to have a perhaps better predict PK in terms of what's going on and relate that to sort of PD endpoints. That, I think, in the preclinical space, and then perhaps will better translate to less failure in the clinical phase. That's a grand ambition, but I think each year there seem to be improvements in the modelling, and a reduction in the number of assumptions made, and a sort of moving away from the black box fear to, but this is actually producing something useful. I think that can only help us.
So in silico modeling, obviously, that's one of the topics that pops up from time to time at conferences, a little bit about drug delivery to the lung, DDL, of which you are a chairman. Where do you think is going to be, in which direction is DDL going to evolve over the next few years? And what are your hopes for the DDL 2026?
Okay. So we recognize that we need to move past running a conference, which is quite easy on just the devices and the sort of new molecules, because as we've talked about already, the people who are driving these inventions, these new developments, are changing. And so one of the things we want to do, as in parallel, if you like, with providing technical forum for the young scientists, is to explore how innovation works. And we ran a pilot session last year, which I think was quite successful, to talk about some of the experiences of people in the inhaled space. They've set up companies, those have perhaps worked or perhaps haven't worked, but why? What was your experience? To try and build an understanding, if you like, in the audience of how some of these smaller companies appear and perhaps disappear. What's the reason? Because more and more, I think those will be the employers of the future. Those will be where the work is done. And I think more having a better understanding of how are these things funded? How do they work? Who drives them? How is success measured in the short term? Can only be a good thing, because I think it links back to what we discussed, a bit of a resilience about change. I think the more you understand some of this innovation process, and perhaps feel emboldened to start up a company or join an early startup, eyes wide open to the risk, I think can only be a good thing. So that's a direction that we're hoping to pursue. And in part because if we can show that we have this interest in how innovation works, perhaps broaden the reach of DDL to a bigger group of people, investors. I've worked with a number of groups who are spending considerable sums of money on developing new inhaled products, never heard of DDL. And you think, but why not? And so if we can encourage the people who perhaps have the funding and the desire to support such activities with the people who have the technical knowledge and the interest and the ideas, that I think might help develop DDL for the next 10, 15, 20 years, whatever it takes.
I think DDL 25 is the first time that I saw on the delegates list a VC company having two delegates, if I'm not wrong. Maybe there were two groups. Am I right? This first time someone actually turned
Yes,
up?
because typically the clientele for our conferences have been people like us
Mm,
who are interested in the nuts and bolts of how
the
to make a
science, yes.
product. Now it is changing.
You did mention something earlier that the industry has changed. In the past, there used to be the big pharma companies doing everything in-house, investing a lot in technology, and they will be sending busloads of of delegates to the conference. And DDL is still the number one conference for our industry, which is about twice the size of the nearest competitor in North America. So clearly it's successful, but something I've observed is this is not the place where the new molecules are. The new molecules, Bio-Europe, Bio-International,
Mm.
PODD, this is where the excitement is, where the deals are made, the molecules are, and there are quite a lot. So what can be done to bring those new molecules which are at a very early phase? Sometimes they are preclinicals, that might be, with a bit of luck, phase one. What can we do to bring them into DDL and say this is where it can be facilitated?
Again, it's a good question. We've debated this a lot recently in the committee to how do we have visibility to the people who are going to those meetings? And in part, this drive to sort of understand better how innovation works, we're looking to set up areas where companies can come in and perhaps present. We've always been quite strict on having quite a lot of data to present, but it may be that we should explore taking more risk,
Mm.
having projects coming at a much earlier stage and talk about their aspirations. And then if potentially you've got backers of projects there as well, that could be a good thing. I mean, we don't want to become a pod or a Buy Europe or something. The idea is we're not trying to copy that. Our thinking is how do we best serve the DDL community in creating opportunities? Clearly, we have lots of good science to present, but equally, if we don't try and match and we're trying to put a lot of time and effort into developing the young researchers, but they need to have somewhere to go. And so we were hoping to see if we could perhaps raise the visibility of the conference to a wider audience, as that may create more career opportunities downstream for them.
So you haven't asked me the question, so I'm going to ask it to myself and answer it. Should you invite companies who are developing molecules for inhalation drug delivery but are not quite doing it yet? So that's my question. The answer is yes. And I would like to tell the DDL committee via you, there are a lot of beautiful molecules out there, which of course are not using drug delivery technology just yet, but are developing, those companies are developing molecules for treating lung disease or lung diseases that could be treated via the lungs. And I think we need to take a bet and invite them.
Mm.
And focusing on molecules, which is very chemistry based, not technology, like not inhalation drug delivery technology. But that I've got this feeling that would revitalize our area by bringing the molecules in and start making people dream about the future therapies we could be developing.
Yeah, I like that idea. We have to broaden our view because it can't be just the simple mechanics of what excipients we use, what bit of plastic do we do. It is bigger than that already. You've seen this. We try and go from sort of the early research on how to sort of build formulations way in advance of any preclinical work starting through to reporting on clinical trials. But there is an opportunity there to highlight interesting new mechanisms, new indications, what might be required. I think to sort of shine a light on the possibles that could come through can only help because if we don't do that, then of course we run the terrible risk of being very complacent. And I think the success that we've had will just flatline and then we
Mm.
may disappear, which would be a horrible thing because I do think the committee does a great job in supporting the youngsters. And so if we, I probably shouldn't call them youngsters, the young researchers. And so it would be nice to think we're putting some longevity into that concept, way past the current committee, who are all, of course, mostly elderly.
Let's rewind 30 years. Young Gary goes into the lab for the first time. He decides to do a PhD. What topic for your PhD would you tackle today? What challenge would you give yourself? You're starting a new PhD and you think, this is it. This is my research in 2026. What would it be?
That is a question, Philippe.
Yes, it is a trick one. Yes,
it is. It really is. I can only answer as I feel today. I think it would be. It goes back to the lung cancer situation. I think it would be right. So we have the background in knowing how we can deliver some of these drugs. How do we improve the targeting? How do we improve perhaps the release pattern to not only once we've gone to the effort of getting the therapeutic entity to the right place, but we maximize its residence time to deliver efficacy, but without having some other tox. I think I would really be interested in looking at really pushing, maximizing what we can get out of some of the current known molecules. And then from that, hopefully learn how we would better deliver some of the newer, hopefully better molecules, or combinations of molecules, because these things are never, as I understand it, this isn't a monotherapy. This is different approaches to try and tackle the disease state. And so if there's an opportunity then to sort of perhaps maximize the sort of what you're hitting the tumor with through different approaches, that's what would interest me. That would, at this moment in time, fire me up.
Great. Well, that will be the topic of your dissertation. Gary, thank you so much for joining me today.
It was a pleasure. Thank you.
And looking forward to many more years of watching DDL and your work in the industry. And we'll meet again.
Sounds good. Thank you.
Thank you.